Assalamualaikum and a very good morning to all participants from Malaysia, the SEAMEO network and all affiliated countries. Welcome to IMR-SEAMEO Webinar Series 2024. The theme of today's webinar is Influenza Surveillance in the Genomic Era, Enhancing Public Health Responses Through Gene Sequencing. And my name is Dr. Nabila. I'm a clinical microbiologist from Virology and I will be the moderator for today's sessions. Before we jump right into our speakers, I would like to use this opportunity today to introduce Virology unit and the activities that carry out in our unit. Over the years, Virology unit has been working on many medically important viruses, and one of it is influenza virus, which is the virus of concern today. In 1954, in fact, the first isolated influenza A virus was isolated from a patient's throat washing in Virology IMR. And in 1966, Virology unit has been appointed by WHO as the National Influenza Centers. And since then, we've been actively participating in the global influenza surveillance and response system. And not only that, we are also act as the National Poliovirus Laboratory since 1998. And in 2010, Virology was accredited and entrusted by the WHO to perform the vaccine intratypic differentiation testing, ITD testing, and also testing for vaccine-derived poliovirus by the WHO. We are also the National AIDS Reference Laboratory, NARL, where we provide consultation for difficult HIV cases, and also we help in screening of babies who are born to the HIV positive mother. And not only that, we also provide external quality assessment that is known as NICAS, National External Quality Assessment, for serology testing involving hepatitis and HIV, and we offer it to many laboratories all over Malaysia. So besides being reference lab or the surveillance laboratory, we Virology are also very committed in conducting highly impactful research pertaining to the medically important viruses such as Zika, dengue, chikungunya, Nipah virus, SARS-CoV-2, respiratory syncytial virus, and many, many more. And we are also helping the Ministry of Health as well as the public health sectors in outbreak investigation and management. And I think the closest example is the recent COVID-19 pandemic. We also involved in the 2009 pandemic of H1N1, and we also assist in outbreak investigation of cluster virus outbreak that happened over the years all over Malaysia. And besides that, we also provide training, and it involves training of the postgraduate candidates such as Diploma of Medical Microbiology, DMM, DAPNE. We also provide training for Master of Pathology, Medical Microbiology, and there are also some candidates of such specialty trainings for medical specialists that were doing training in our lab. We hope that our unit will continue to advance and expand its contribution within the medical field by also involved in development of testing kits and also development of important vaccines. We aim to bring upon a positive impact to the society as a whole, not only to the medical field but to the public as a whole. And being here today, by participating in the webinars today, it is also a way of connecting the virology to the public, to the society, and to educate the society on the virology medical aspect. Ladies and gentlemen, this webinar series was initiated with the aim before we begin the presentation, allow me to announce a few housekeeping messages that I received from the Secretariat. First, we will not be taking questions during the presentation, but participants may post their questions using the link shared in the YouTube channel. Second, the questions and answer session, the Q&A session, will be conducted after both presentation have ended. And there's also a attendance link in the YouTube channel for those who like to have the e-certificate of attendance. And lastly, please subscribe to our National Institute of Health Malaysia and NIH Malaysia YouTube channel and click for notifications so that you can be notified when there's a new post. So ladies and gentlemen, we are fortunate today to have two distinguished speakers with us. Allow me to introduce our first speaker, Dr. Banusha Vanukuppal. She's a medical officer who's in charge of molecular diagnostic and molecular laboratory in virology. She's been actively involved in studies and research on respiratory viral infection, including influenza. And today, Dr. Banusha will be sharing her valuable experience with a presentation titled Unraveling Flu, Basic Science and Surveillance Strategies in Influenza Control. Dr. Banusha, I'm going to pass the baton to you. The screen is yours.
Thank you so much for the kind introduction, Dr. Nabila, and very good morning to those who are attending the section today. So I'll be basically unraveling flu, the basic science and surveillance strategies in influenza control. So before we go further, we have to revisit the history. We have to know the deep, dark history of influenza, which happens in 1918 after the World War I, where literally 1/3 of the world population died because of this flu. So it's about 6,000 million deaths from influenza happened in 1918, and how we would have curbed at that time when there is not much technology or pharmaceutical availability was available at that time. As you can see in this picture, it kind of reminded our quarantine centers during the COVID pandemic as well. So there are mitigation measures, there were prevention measures were taken even at that time. However, the current disease burden is still there. There are around 1 billion cases of seasonal influenza annually, including 3 to 5 million cases of severe illness. It causes about 290,000 to 650,000 respiratory deaths annually. 99% of deaths in children under 5 and with influenza-related lower respiratory tract infections are in developing countries. So before going further on this deadly virus, we have to know the basic science. So the viral property and classification of influenza, it belongs to the Otomyxoviridae family, and there are about actually 7 serotypes under the Otomyxoviridae, but there are 4 types which are major human pathogen, which are A, B, C, and D. These are negative segmented RNA genome, and it contains about 10 major proteins, but mostly 8 major proteins are the one we are much concerned about. Other than that, there are 2 major surface protein of A and B viruses, which is we're going to talk a lot more after this, which is our hemagglutinin, which I'll mention as HA, and also neuraminase, which I'll mention as NA. So influenza A virus is identified by the serotype of the 2 major surface proteins. As I mentioned, there's about 1 to 11 types of NA and 1 to 18 types of HA. And for influenza B, we won't be actually using the subtype of HA and NA, but it has the similar structure. We can see the bilipid envelope, the viral RNA, NA, and HA as well. And there's also ion channel, the M2. So influenza virus genome, as I mentioned, there's 8 more important genomes, which is PB2. It works in virus replication, PB1 in virus replication and apoptosis of the cell, PA for virus replication, HA for the virus attachment, and antigenic determinant subtype specific, which is H1 to H16, NP for RNA coating and nuclear targeting, NA for virus release from the cell, antigenic determinant, as well as subtype specific. For M1 and M2, it's the membrane protein, and NS1 and NS2 is also known as non-structural protein. So these are the main 8 genome of the influenza. This was based on the influenza of H1N1 found in 2009 pandemic. So what influenza A and B causes? We can say influenza A is much potent compared to influenza B. We'll go to that after this, but we have to know influenza A actually can cause seasonal influenza. It also causes zoonotic variant influenza in the animal species, and also it actually can cause pandemic. For influenza B, mostly it causes about sporadic outbreaks, and sometimes even pandemic happens. So causes of epidemic usually happens in winter temperate countries compared to tropical countries. We don't usually see obvious seasonal variation, but it's there. Meanwhile, for influenza A virus, as mentioned, it affects humans, birds and other animals. B, mainly infects the humans. C, it causes milder disease in humans. And influenza D, it causes infection primarily in the cattle, the cows and all. So influenza A virus subtype, the basic reservoir is aquatic birds for all the HA and NA subtypes. So that will be the main reservoir. However, we also can find the virus in poultry, humans, pigs, horses, aquatic mammals, cats, bats, and even dogs. So these are the known strains according to the animals. For humans, the most known strain is H1N1, H2N2, H3N3. In bats, it's H17N10. In swines, it's H1N1, H1N2, H3N2, H4N6. In horses, it's H7 and N7, and H4N8. Meanwhile, for aquatic animals, there's H7N7, H4N5, H3N2, H3N and H13N9, and poultry will be H4, H5, H7, H9. It's like basically the birds, we can find all the HA and N in them, okay? So the evolution of influenza virus can happen due to many reasons. It could be segmented level. There could be mutation due to RNA-dependent RNA polymerase, where there's one mutation per genome replication. There could be codon insert deletion indels, where it occurs in HA, NA, and NS genome. Non-homologous recombination, for example, the HA cleavage site of H7N3 with either NP or NM. That means it will be substituted. And there could be even selection pressure between the viruses. And most importantly, antigenic drift in HA and NA. This commonly happens in influenza A, which I'll go through in the next slide. And the drift, it could be because of the host or the geography of the places. I mean, due to geography as well. So what is antigenic drift? Antigenic drift is actually a minor mutation occurs in the HA and NA during viral replication. It results in new variants which is partially neutralized by our previously acquired antibody from the last infection. Meanwhile, antigenic shift, there will be an abrupt major change in flu A virus resulting in new HA or NA proteins in the flu virus that infects human. And this shift usually results in new flu subtype infecting people for the same time. And basically, antigenic shift is the most common cause of a new pandemic occurring. So the viral transmission, it can happen via direct transmission, that means from the reservoir straight to the human. It could be due to adaptation from another animal to another animal, and also due to reassortment. Reassortment I'll go through in the next slide, but to understand it will be like from different species of animals or human, and then it could be going into another animal and then infecting human again. So the timeline emergence of human influenza viruses, it actually happened in 1819 Russian pandemic, where there was circulating strain of H3 and N8. Later in 1918, the major one happens during this post-World War I Spanish pandemic H1N1. And 1933, we already see there's a circulation of H1N1. In 1942, that's when we actually discovered influenza B. And then in 1957, there were avian influenza, the replacement of H1N1 with H2N2, and it causes Asian pandemic. In 1968, there's Hong Kong pandemic by H3N2 through swine flu. And in H1N1, there's children pandemic, happens in 1977. And then in 1987 and 1988, there were a pandemic caused by influenza B, B Yamagata and B Victoria together. In 1977, there were H5N1 by avian flu, H5 avian flu. And then 2005, again, we found this 1918 sequence where we found from the Spanish pandemic occurring here. And in 2009, where the latest, where we all experienced, there were H1N1 pandemic, even Malaysia experienced it, and IMR was like involved. It's via the swine. And in 2013, there was another avian flu outbreak, pandemic, sorry, H7 and N9. So there's like circulation of influenza A was found, and there was co-circulation. Meanwhile, for influenza B, there was co-circulation of both the lineage throughout since 1891 until 2018. So this is basically the timeline of major pandemics happening. And we can see here the resortment history of human pandemic of influenza viruses. In H1N1, there was all, if, for your understanding, all were blue. Then in H2N2 1957 Asian flu, we can see there's avian and also there's mixture of Spanish flu found. In H2N2 1968 Hong Kong flu, we can see there's a mixture of avian influenza and also human influenza together from the Asian flu. And in for the H1N1 in 1977 Russian flu, they actually suspected there could be a possible escape from laboratory freezer where the same gene was found again. And for H1N1, the current latest pandemic of influenza 2009, we can see there's actually mixture from the avian influenza, the previous influenzas, the swine influenzas, and also, it was found in human at that time. So human seasonal influenza virus is, as we know, A and B, can be subtyped. For A, we subtype according to their H and N, and there's clades and also subclades. Meanwhile, for influenza B, we call it via their lineage, which is B Victoria and B Yamagata. So for WHO influenza virus nomenclature, when we can see in the websites, we basically call them according to influenza type A, mentioned here, and then the place of isolation, the year it has been isolated, the isolation number, and we actually mentioned their hemagglutinin HA and NA site. And this goes for influenza B. So for influenza B, we go via lineage because it's only found in human. And I actually wanted to show this particular picture because we had influenza B Lee in 1940, and after that it was not found. Then we had influenza B Russia in 1969. After that it was not found. Currently, we have circulation of B Victoria, and actually B Yamagata was not being circulating for some time after the COVID pandemic, but that's the dates. Okay, currently why we are actually more concerned of influenza in Malaysia because of the symptom and complication it actually causes to the humans. For the symptoms, it can actually causes neurological symptoms such as fever, headache, confusion, respiratory symptoms such as dry cough, sore throat, nasal congestion, gastrointestinal symptoms such as nausea, vomiting, diarrhea, and musculoskeletal symptoms such as myalgia and fatigue. However, this can be actually become a bigger complication, especially those who have lower immune system or immature immune system or have lower immune defense. For example, in kids or the elderly or people who have been immunocompromised. For example, the complication from neurologic could be febrile convulsion. For in cardiac, it could causes pericarditis, myocarditis. Respiratory, it could cause pneumonia. Pregnancy, it actually causes smaller neonatal size, increased maternal complication, and musculoskeletal wise, it even causes myositis or rhabdomyolysis. And all these complications are more found, as I mentioned, in children and even in elderly. So for laboratory diagnosis, there are few types of method that we can actually use to detect this virus. We have antigen detection. We use immunofluorescence. We have molecular tests, real-time PCR method. We have serological tests. We can use hemagglutinin inhibition, HI test, complement fixations. We have test neutralization, and etc. And we actually can do viral isolation using specific cells like kidney, MDCK cells, and LLC MK2. So for treatment-wise, as advised by CDC, we have few, which is commonly used in Malaysia, is Tamiflu now, which also known as oseltamivir, for patients more than 2 weeks old. It is formulated via oral. And we also have Zanamivir for patients more than 7 years old, inhaled powder. Peramivir, we have IV dose, and before Balozawi, we have oral dose. And this can be given based on the patient's age, as mentioned. And most importantly, the prevention is better than cure, as we say, vaccine. So in March or April, we have vaccine formulated for Southern Hemisphere, and another one will be in September, October for Northern Hemisphere. There are two types of vaccine: inactivated subunit split vaccine given by injection, or we can even give live given by intranasal. So there will be an annual reformulation of influenza vaccine made annually based on the current circulating strain in Northern and Southern Hemisphere. And the immunity will be for 1 year. So the vaccine options available will be a cell-based flu shot, which contains virus grown in cell culture. We have a recombinant flu shot, which is completely egg-free flu shot. We have egg-based high-dose flu shot, which is approved for use for elderly patients. We have egg-based adjuvanted flu shots and an egg-based live attenuated flu nasal spray vaccine as mentioned. So some vaccine options available in Malaysia is egg-based. We have Fluarixi Tetra. It's an inactivated influenza vaccine. We have inactivated influenza vaccine, flu quadrivalent vaccine, Vaccine-Grip Tetra, and also Sky Cell Flu Quadrivalent, which is a cell-based influenza vaccine. So coming to surveillance. Influenza surveillance in Malaysia, the sourcing patients at Sentinel site, we have about 18 SARI Sentinel site and 41 ELI Sentinel site for Malaysia. And for IMR wise, we actually receive more than 90 samples per week. We have about 18 Sentinel sites as mentioned at the site. And the surveillance activity, it actually happens daily, and the report will be done weekly. That means we'll be receiving samples from all the 18 Sentinel hospitals daily, and we will do the tests on the samples, and we actually will be reporting to the Surveillance Section Disease Control Division under KKM Malaysia, and also at the same time to WHO Collaborating Center, directly to WHO centers. So for WHO wise, we have Global Influenza Surveillance and Response System, where WHO tracks seasonal influenza virus and makes recommendations for vaccine through a large global network called GRIS. This network is also monitor other influenza virus such as SARS-CoV-2 and RSV that could be pandemic virus. It therefore acts as an alert mechanism as well, and GRIS also strengthens laboratory surveillance to prepare for and respond to outbreaks. Together with GRIS, we also have PIP framework, which is known as Pandemic Influenza Preparedness framework, which brings together stakeholders to implement a global approach to pandemic influenza preparedness and response. It has, for example, signed agreement that have secured 10% of vaccine production, over 400 million doses in the event of a pandemic. And this is for the worldwide response. Coming to the surveillance, actually under WHO and GRIS, we can actually view the influenza laboratory surveillance information real time. So I'm just going to go to the website and show you guys. So this is the website, we call it as FluNet. So here we can actually view the surveillance being done worldwide in real time. So you can actually choose any country you want to see how's the flu being monitored there according to your region and influenza transmission zone, the hemisphere, and also surveillance type. So for example, now Malaysia, we are under West Pacific region. We can see what are the current circulating flu viruses according to the year and time. So, and we can go to Malaysia. Sorry. Okay, so this is our current circulating viruses found in Malaysia at this time. And we can see this in real time, and these are the data being given by our KKM. Okay, I think that's all from my presentation that I would like to mention. I would like to say thank you very much for the opportunity given, and I will pass back the section to Dr. Nabila. Thank you.
Okay, thank you, Dr. Banusha, for delivering such an intriguing talk. If anyone has questions regarding the topic of the presentation, please feel free to submit the questions in the link provided in our YouTube channel. From the presentation, we know that influenza virus is not a new virus, isn't it? It has been circulating on the earth since decades ago. But one fascinating aspect that I would like to emphasize, which I think is interesting, is that we're not only looking at the evolution of the influenza, but we can also witness the evolution of the medical virology field. Why do I say so? Is if you look at the early years, the detection of the virus or identification of the virus is made through the viral culture on the animal cell lines, for example, and also using this complement fixation testing or heme agglutination inhibition testings, and then it moves to the antigen detections, either using monoclonal antibodies or immunofluorescence testing, and then it moves again towards the nucleic acid detections of this virus, also more commonly known as PCR. And now we are entering to another advanced phase where we are using viral genomic sequencing for virus studies. And when we're delving into viral genomic sequencing, there is a lot more to know about the viral genomic sequencing. And for that purpose, we would like to invite our second speaker, Dr. Khayri Azizi Kamel, our research medical officer. He is actively working on viral genome sequencing, especially on SARS-CoV-2 virus as well as influenza virus. His presentation title is Application of Nanopore Sequencing Technology for Influenza Surveillance. Dr. Khayri, you ready? So yeah, over to you, Dr. Khayri. The screen is yours.
Thank you very much, Dr. Nabila, for the kind and warm introduction. So before I begin, let's just have a quick look through the journey of how the history of DNA sequencing evolved. So it begins in 1953, where Crick, Watson, and Franklin they discovered the double helix structure of DNA. And in 1977, Sanger sequencing, which is currently the gold standard of sequencing technology, was developed. And this was actually used to sequence the complete genome of 5X, which is a type of bacteriophage. The sequence was a total of slightly above 5 megabases. And in 1998, this was the era of NGS or next generation sequencing. Next being next after Sanger was developed. The principal technology was called as SBS or sequencing by synthesis. This technology actually underlies many of the current sequencing technology. This technology underlies many of the current sequences that are considered as NGS or short-read sequences. And when we compare Sanger with NGS, Sanger actually was used to previously sequence human genome, which took 13 years, as opposed to the NGS, which we can use to sequence human genome in a matter of hours or days. And in 2011 and 2012, this was when the long-read sequences or third generation sequences was being introduced. And one of them being the nanopore sequencing technology, which is our talk for today. So sequences are generally divided into generations. So these usually depends on the time that they are being introduced. So the Sanger sequencing technology, as we've discussed before, this was followed by the NGS or second generation sequencers. The beauty of the NGS or second generation sequencer is that it started to introduce the concept of parallelization or high throughput sequencing. And this was why we can now sequence human genome in a matter of hours or days as opposed to years. And later on, third generation sequences were introduced, Nanopore being one of them, another being PacBio. This actually helped with the assembly of the sequencing reads because these are considered as long-read sequencing machines. So you would have a better structural accuracy compared to short-read sequences. So what about the history of how nanopore sequencing technology was actually proposed? So the idea came to light in 1989 when David Demer, at that time, was driving, and he came across the idea of how we can actually sequence DNA strand through nanopore. And this was when he was driving, actually. And he worked later with Church and Brenton, and subsequently they patented this idea in 1998. And eventually, in around 2012, the first nanopore sequencing machine, which is the size of probably 4 to 5 times the size of our USB flash drive, is being introduced. Its early application in within the realm of virology, I think we can see how it benefits during the Western African Ebola virus epidemic in during 2013 and 2016. And this was when around 2016, where Joshua Quick, who was at that time he was a postdoc working under Professor Nick Lohman lab at University of Birmingham, actually went on to develop a mobile lab in the Western African region, and they make use of the portability of the nanopore sequences to be able to sequence and carry out the surveillance of the Ebola virus throughout the epidemic. So what are the advantages of nanopore technology when used in pathogen outbreak surveillance? So as I've mentioned, and we can see that it's portable, and with low startup costs, that means that it's suitable for decentralization. We can see the example in Ebola virus where instead of these viruses being sent to probably sequencing lab to other countries, this can actually be done within the Western African region themselves. Second, the sequencing run can be stopped and flow cells can be washed and reused if enough data is obtained. So the second point, it follow up with the low startup cost means that there is an advantage there in terms of the cost as you can wash and reuse, so there is a flexibility there. Real-time analysis. So analysis can actually begin as during the run as the data is being generated. There is no need to wait for the sequencing run to complete. You can begin to analyze. And if we feel that enough data is obtained, then you can actually stop the run and reuse the flow cell for subsequent run. Now, the fourth point, I think it's more from my point of view. I think it's interesting that with this, with nanopore technology, actually, in case of there is error, power supply trip, etc., the sequencing run can actually be restarted. And I think this is as important during an outbreak that happen because a lot of uncertainties sometimes occur. So we see here some of the sequencing technology that was used during the COVID-19 pandemic. So majority of these are by Illumina. So Illumina being the second generation of next generation sequencing, followed by nanopore sequencing technology. But I want to highlight one thing interesting here. Nearly half of the sequencing technology that is being used in low-income countries actually use the third-generation sequencing. So, and the third-generation sequencing being most of them, we can deduce being the nanopore sequencing technology. So, again, it highlights how this technology can help to decentralize sequencing tests, and so provide a better surveillance strategy during pandemic. This is an example of how we actually use the nanopore sequencing in our lab during the VOC Omicron outbreak. which started end of 2021. So, as I mentioned, again, after we've actually extracted the viral RNA and then proceed with library preparation, sequencing and base calling are done. And as highlighted before, we can analyze the data as the run is being as the run is being performed. So it took us about I think about 3 hours, if I'm not mistaken, that where we sort of had an idea this is probably the first VOC Omicron case. We waited for a few more hours just to have a little bit more data. And finally, after 6 hours of sequencing, we actually stopped the run and confirmed our first VOC Omicron case in Malaysia. This was on the 2nd of December, 2021. So moving to influenza viruses, we had an opportunity, our team in August 2022, to attend the International Influenza and SARS-CoV-2 genetic sequencing course. This was organized by the Ministry of Public Health of Thailand, along with US CDC and the American Public Health Labs as well. It was a one-week course where we learned to sequence both COVID and flu, and we were grateful for the opportunity to attend the workshop. So what are the objectives of sequencing flu and performing surveillance? So 4 points here that I want to highlight. The first being to monitor the evolutionary changes of the virus. As you've heard from our speaker earlier, Dr. Banusha, she has mentioned that there are various subtypes of flu viruses. So it's good to actually monitor how this change can contribute to the development of diagnostic assays. So a lot of our PCR tests, even our rapid tests, these are being developed from the sequences that are generated from sequencing and surveillance of influenza virus. The third one, this is a very important one, is determining the seasonal influenza vaccine composition. And the final one is to identify genetic changes associated with antiviral resistance, as we get an info of the whole genome of the flu virus. So a bit of our workflow of how we do sequencing of flu virus. So usually it will take us about 4.5 days. We recently tried a rapid method, so this can be shortened to 3.5 days. So the first step actually involved the extraction and real-time PCR. These are mainly from screening of these samples, and we proceed with sequencing for samples with CT value of 30 and below. So before I move on to the sequencing itself step, allow me to actually highlight again that with the influenza virus genome, there are 8 segments. So when we say that we would like to sequence the whole genome of flu virus, that means that we will need to sequence the whole 8 segments of this genome. Interesting to these segments, you can see at the ends, there are 12 or 13 nucleotides which are considered conserved bases. So we can actually make use of this to design primers and subsequently to amplify the whole 8 segments of the influenza virus genome. This was highlighted in a paper by Bin Zhou, and this remained the standard method to amplify the influenza virus genome. So after amplification, an example of a bioanalyzer electropherogram. So I know that I did mention there are 8 segments, but because of some of these segments, the size are quite similar, so they overlap each other. So usually we get about 4 or 5 segments. The picture on the right is the simulated gel electrophoresis if we were to perform one. The library preparation. So the purpose of library preparation is actually to ensure that the segment that we have amplified, these are compatible with our sequencing technology. So we can see there is, so after end preparation, there is a step called barcode ligation. So these actually help to allow many samples to be sequenced at once and so reduce the cost. Last but not least, adapter ligation. We do that to ensure that this segment can be compatible with our nanopore. Sequencing itself. So the machine that we used during our VOC Omicron outbreak was a bit different in the sense that it has a integrated computer, something like a tablet along with it. So you don't need to have another computer to connect to it while it's running. So again, just to highlight again the principle of how nanopore sequencing works, as the DNA strand travels through the nanopore, a disruption of the current is being recorded, and this disruption is unique to each nucleotide basis, and from this is where the sequencing reads are being generated. About half a day after we have sequenced, we proceed with bioinformatic analysis. We're quite fortunate actually because there is an available workflow for us to actually use to analyze this. To actually analyze the sequencing rates. These are available at GitHub. You can use command line, or we actually also have a software, so it's just a drag and drop method. Last but not least, what is the limitation of this technology? The accuracy of base calling is considered less than short read sequencing technology. Having said that, it is catching up. Second, rapid development. This is a make it or break it sort of situation whereby sometimes, updates to softwares can sometimes cause errors, etc. Bioinformatics workflows are not as widely available compared with the data generated by short-read sequencing technology, probably because this is a relatively newer technology. And the fourth point, there is a caveat to washing and reusing flow cells because it depends on several factors such as storage condition, previous run condition, etc. So sometimes, in my case, I sort of consider, the ability to wash and reuse flow cells as a bonus. So before I end, let me say thank you to our team from the Virology unit, and many thanks also to CDC, APHL, and the Ministry of Public Health for the opportunity provided to us to attend the sequencing workshop. I think it has provided a very, I think it has actually been very fruitful, and we have actually able to sequence many of our current circulating strain of influenza viruses. With that, I thank you, and the screen, I pass the screen back to you, Dr. Nabila.
Thank you, Dr. Khayri. It was a very insightful presentation, and I like how Dr. Khayri brought all of us back to the history of sequencing, and we can see the progress of the sequencing itself until it's been applied in our setting. And I believe now that all of us has grasped the significance of viral genome sequencing in influenza surveillance, and we will now be moving to Q&A session. Again, please do not hesitate to send us your questions during using the link provided in the YouTube channel, and I will try my best to connect all of the audience with our distinguished speakers through the posted questions. Nevertheless, should the time does not allow us to address all the questions, our speakers will try their best to answer all of your questions through the email. Yeah, it seems that we have received a few questions from our audience. Okay, let me see. Yeah. Questions. The first question is to Dr. Banusha. The question is, will the vaccines that been distributed in Malaysia covers all the circulating strains? I think it covers all the local circulating strains. Dr. Banusha?
Yeah, thank you so much for the question. Well, the vaccine distributed in Malaysia, basically it does cover according to the southern hemisphere. That means Malaysia is located in West Pacific region and we are considered we are in the southern hemisphere. So as I mentioned before, the WHO will combine all the data given in the JIRS system and they will actually make annual vaccines according to the circulating strain and distribute according to the hemisphere given. So Malaysia is considered in Southern Hemisphere and the vaccine distributed does cover all the circulating strain. Thank you.
Thank you, Dr. Banusha, for the thorough explanations. I think it's answered the questions. And okay, now we have another question. Is to Dr. Khayri. What is the most challenging issue facing while doing influenza genome sequencing? This is from Nurani, UM.
All right. Thank you very much, Nurani. So the most challenging issue facing while doing influenza genome sequencing, I think a lot of our library preparation, the wet lab part has been, I mean, from what I've noticed and our experience, it's becoming much easier. So I think that there are two challenges here. Number one is the data that is being generated. So when you already have, a very simple workflow, then if everybody starts to do a lot of sequencing and you get a lot of this data, and trying to make sense of it and trying to analyze it, I think that is sort of our bottleneck now. And another point that I want to highlight is another issue is sometimes in terms of the sample itself, because I think sometimes we have this expectation that sequencing can actually solve a lot of our questions, but sometimes if we do not have a good quality starting material, then it's going to be very difficult for us downstream, both from the wet lab aspect and also from the bioinformatics analysis as well. Yep.
Thank you, Dr. Khayri, for thorough explanation on the questions, on the answers. Yeah, I believe I agree with Dr. Khayri. There are, of course, challenges when we are doing influenza genome sequencing, and not only in influenza, but all other virus genome sequencing. And yeah, we also had another question. And this question is to Dr. Banusha. Okay, I think this question is quite simple. What is the current circulating strain of influenza virus in Malaysia? Dr. Banusha?
Thank you, Dr. Nabila, and for the question. What is the current circulating strain? Basically, as mentioned before, we have all the flu A and flu B of the human strains are found in Malaysia, but it is like still under control, so there's nothing to worry much. But the circulating strain most commonly for influenza A is H1N1 from the pandemic 2009, and also H3N2 together with influenza B Victoria. We haven't seen Yamagata after COVID pandemic for now. So these are the three main circulating strains we have found in our lab and in Malaysia. Thank you. Back to you, Dr. Nabila.
Yes. Thank you, Dr. Banusha, for the answers. Yeah, he's correct. H1N1 and H3N2 is the most frequent or the most common circulating strain of influenza in Malaysia. Not forgetting influenza B Victoria. Yeah. And then can we move to the next question? Again, this question is to Dr. Khayri. The question is, despite limitations of nanopore sequencing, do you see that one day this technology will replace the previous generation of sequencing machines? Oh, yeah. Dr. Khayri?
Yep.
Okay.
Thank you very much for the question. So I actually don't think that it will be ever replace, even our Sanger, probably still be the gold standard, at least for many years to come. What I've seen is that most of these technologies actually complement each other. Uh, and it is important for us to be wise to actually choose, which technology that you would use to address your research question. And sometimes you can sort of choose this based on your resources as well. There are options nowadays where you can do hybrid sequencing. This is where you perform sequencing with the same sample, but with two different machines, and subsequently you do what is called as hybrid assembly. So you assemble both data from different machine, and in previous, in some research that has been conducted, this actually provide many additional benefits. Yep. Over to you, Dr.
Yeah.
Nabila.
Dr. Khayri, I actually like your statements just now in which you are saying that all of these methods are actually complement each other because when we are doing research or doing surveillance or diagnosis and many more, we have to look at what are the purpose of the testing, whether it's a whole genome, whether it's for detections, whether to look for the molecular characterizations or evolution and all. So this purpose and will actually decide or will actually lead you to which technology or virus sequencings that we would like to use, isn't it?
Yep.
Yeah, I agree with Dr. Khayri. And I think that is the last questions that we have for today. Oh, sorry. Yeah, there's another one. Sorry. I actually received another question. I think this is going to be the last questions. This question is to Dr. Banusha. What is the current laboratory testing method used for influenza surveillance at your center?
Thank you so much, Dr. Nabila, and for the question. So currently, we are using real-time PCR method for detecting influenza in IMR. So for detecting influenza, we have few steps. Although we are using as in the assay that we use. First, we have to detect influenza A or B from the sample sent, and from A and B, we actually the positive ones we will subtype later, and then we will inform to the ministry. So that's how the test actually to culture whether we need to culture the sample using this particular or specialized cells, or at the same time we are currently also started doing the sequencing. So we'll be using the samples to do our genome sequencing as well. So these are the laboratory testing methods currently we use in IMR Virology unit. Thank you. Back to you, Dr. Nabila.
Thank you, Dr. Banusha, for the explanations. I'm afraid that that this is all the time we have for Q&A sessions. Any additional question, do not worry. You can still submit to our link in the YouTube channel, and we will try our best to answer all of the questions through our email. And we hope that this webinar shed light on some research work that we are conducting in IMR and would spark interest and could initiate research collaborations with others. Ladies and gentlemen, now we have come to the end of the webinar session. I would like to thank your support has made this session successful. And don't forget to subscribe to our National Institute of Health Malaysia YouTube channel. And again, don't forget to click notifications so that you will be notified when we have a new post. Before I conclude, I would like also to announce that we will have the next session of IMR-SEAMEO Webinar Series on 31st May 2024. So please mark on your calendar. And the theme for the next webinar is Upholding the Malay Traditional Knowledge of Natural Resources. A very interesting topic. And we hope you can join us at the next sessions. Till we meet again, I will end the session today with Selamat Hari Raya Aidilfitri, Maaf Zahir dan Batin. Take care and stay safe.